Hello, thank you for joining us today. We have a very special program featuring scientists and doctors and also industry experts. The panel today is called Who is the Patient and How Do We Make Sense of All the Data We Collect? From genes to social context and life habits, modern technology allows us to gather an immense amount of data on each patient. Here, we will discuss how to use data to characterize relevant populations, how to make sense of all the data collected on patients, and how to balance precision and statistical significance. Feel free to ask questions in the comment section on YouTube. On the panel, we have Phillipe Nore, Vice President of the International Market Development for clonoSEQ at Adaptive Biotechnologies, Marina Sirota, Principal Investigator and Associate Professor at the Bakar Computational Health Sciences Institute at UCSF, and Dr. Cristina Lanata, an MD and Assistant Professor of Medicine in the Division of Rheumatology. Thank you all for joining. So let's first talk about the need for data in healthcare. Cristina, you are a rheumatologist and your research is focused on population-based genomics to better understand SLE. From what I understand, SLE stands for systematic lupus erythematosus. First, did I say that right? And second, what is SLE? Also, could you tell us how doctors use data to characterize relevant populations?
Thank you for this wonderful invitation and good morning, everyone. I always love to speak about lupus, which is a very confusing disease starting by its name. It's called systemic lupus erythematosus, or SLE. And it's a disease that I've had been very passionate about since med school. It's a chronic autoimmune disease. It affects mostly women. Out of 10 people, 9 are women, mostly in the reproductive age. And it's characterized by the production of autoantibodies. So the immune system gets sick and generates a whole set of immune mechanisms that generate a lot of what we call immune complexes, and these land in different organs and then cause disease and damage. So it's a disease that can look very different. I'm from Peru, and the disease I saw back home was very different than the disease I see here, sometimes in different areas of the US since I've been here. And it really is an example of how heterogeneous one disease can look like. And I was very drawn upon what makes the disease so different, just based on my own personal experience of seeing people from different ethnicities and different backgrounds having such a different manifestations. Right now, we have what we call a clinical diagnostic tool. So how do we call a patient lupus? Is we have a set of criteria that we've been observing through the years, but they're mostly clinical criteria. And then we've developed 11, and then for both research purposes, really, but the diagnosis really made clinically, but we can say that someone has lupus if we find a combination of 4 out of 11 of these classification or diagnostic criteria. So as you can imagine, one patient that has a diagnosis of lupus can look very, very different from someone that has a complete set of other 4 clinical criteria, which makes it very, very complex to study and understand this disease as a whole. It's one of the reasons that we have so many clinical trials fail in lupus. It's because we might be treating a bunch of different subtypes, all together. So there's really an unmet need of trying to really understand this disease and how different it can present.
And you're also a doctor who interfaces with patients regularly. From your perspective, how can patients benefit from big data?
Yeah, so big data is such a big term, but I think it speaks to we really need more tools. And I think as technology goes forward, we are being able to understand and describe biology in an unprecedented way. And I think that's where we are right now. We're being able to process not only genetics, but gene expression, chromatin structure or epigenetics, and also just a lot of just proteins in serum, proteins in urine, and also just different ways of actually profiling the immune cells, including mass spectrometry. And so there's like a wealth, and every year there's like a different technology that comes out what measures a different part of gene regulation or gene expression, and it really is exciting, and it just really keeps growing. I think overall, as I mentioned, it would be really wonderful to have, instead of going from clinical criteria, which is also very important, to molecular criteria, and that's the immunologic criteria, to actually really understand what's driving disease pathogenesis and offer patients treatments appropriate to what they have. And a good example, for example, is cancer. Right now in lupus is, for example, we would be treating all cancer patients with the same chemotherapeutic agent. And that is where we are in lupus. So in cancer, now we have so many different molecular biomarkers to really target specific chemotherapeutic agents to the specific cancer. And I think that's where big data can help rheumatology and autoimmune diseases in particular.
Excellent, excellent. And Phillipe, so you're an industry expert. Can you tell us about adaptive biotechnologies and how the company meets the needs of the patients?
Absolutely, and thanks again for having me. Adaptive is an immune medicine company. So what does it mean? We're very focused on studying the immune system and learn how it naturally detects and fights a wide range of diseases. And the way it does that, the information on how it does that is held within the genetic code of the B cell and T cell receptors that we all have and that are a big part of the immune system. So we have developed a platform to decode that genetic code and translate that into data. And then that data is used to develop diagnostic tests or therapeutics. That's how we learn how the immune system detects and fights diseases. And again, it has a lot of applications across multiple diseases because our immune system is here to fight all diseases that we come across. We always say that every person's immune system has a story to tell because they're slightly different, and that's what we focus on. And we can do that also at a population level and learn a lot. I think a good example and a recent example is our work around COVID-19. I think we have all read about the role of antibodies when it comes to COVID-19, but it turns out that the T cell part of our immune system is equally important. We are facing a question such as, why does someone get sick and die when someone else will not have symptoms? We are now facing questions about how long the immunity will last for people who have been sick or people who get vaccinated. And that's the type of answers that we're trying to provide by collecting thousands of samples of patients and decoding them and really trying to help scientists within adaptive and outside of the community, because we publish a lot of data publicly, help them understand the immune response to COVID-19 and how immunity will work for patients, again, who got exposed to the virus or got vaccinated. And antibodies alone cannot provide this answer. The cell response starts earlier, even before when symptoms are here. So it's a good way to detect the disease early. And they last a lot longer than antibodies. So that's why we think that is a very complementary approach to a problem such as COVID-19 compared to just looking at antibodies.
That's really interesting because you're right. We've only been hearing about antibodies, and now we're just looking into T cells. That's a good point. So Marina, can you tell us about the work being done at your institute, and how do computational scientists approach these problems?
Absolutely. First, again, thank you for inviting me to be part of this panel. And I can tell you a little bit about my work as a computational biologist. So I'm trained both in computer science and biology. And what I try to do in my group is analyze different types of data, whether we use clinical data, so the type of data that Cristina was just talking about, or molecular measurements, such as the type of data that Phillipe was discussing. we really want to see how we can aggregate this type of information, integrate it using statistics or bioinformatics or machine learning, depending on how you'd like to call it, and ask new questions about either disease diagnostics or therapeutics. And there are many challenges with using and analyzing data. Some of those relate to having good annotation. Some of them relate to data heterogeneity. But there are statistical approaches to tackle those problems. Specifically in the context of COVID-19, one of the areas that my group has focused on is computational drug repurposing, or the idea that we can take drugs that are already FDA approved for a certain indication, for a certain disease, and see if we can identify new uses for those compounds. And we can do that with different types of data. You can imagine leveraging molecular measurements. So for instance, gene expression is an example. Gene expression is whether a certain gene is turned on or turned off. And we can look at which genes are turned on and turned off in a disease. And we also can see, based on molecular measurements, which genes are turned on and which genes are turned off by a certain drug. And our hypothesis was, if we take the two, the disease data and the drug data, and we combine them, looking for drugs that might reverse the genes that are affected by the disease back to normal, then maybe we can identify new therapeutics. So the preprint that was just on the screen a few minutes ago is our work on COVID-19, and we were able to do this type of analysis completely using data from the public domain. You might know that pretty much every study that's funded by NIH, we have to, whoever generates the data needs to put the data in the public domain. And that really creates an incredible opportunity for computational scientists such as myself to ask questions. So our analysis of COVID-19 data is completely based on data that's free, and anybody, any one of you, can go online, download those data, and if you know a little bit of statistics and can program just a little bit, doesn't have to be a lot, you can start analyzing those data sets. And that's what we've done there. We've identified 25 candidate drugs for COVID-19. We tested 16 of them with various collaborators because my lab is all computational, just computers. We worked with Melanie Ott's group at UCSF and also Adolfo Garcia Satra's group at Mount Sinai to actually validate some of these drugs in cell lines. And we saw that out of 16 that we were able to validate, 11 of them showed antiviral activity. So this is an example of taking free data that's just out there, patient data. Some of these data sets came from patients with COVID-19, looking at which genes are turned off and turned on. And we were able to make a number of hypotheses and then test them in the lab. And this is really the framework that I see for computational analysis, where you can take a data set, generate some hypotheses, and work with either clinicians or wet lab scientists to actually test them in the lab and see if we can move biology and medicine forward.
Excellent. Yes, that's right. We do need more patient data, and I love that you've been working with Melanie Ott and Adolfo, as they are both a part of the QCRG, which stands for the QBI Coronavirus Research Group. It's excellent that you're working with them, and look forward to hearing more about that. And besides lupus and COVID-19, let's be disease agnostic for a moment. And yes, we will be covering COVID-19 some more, but let's take a step back and just talk about the immune system in general. So Cristina, what can you tell us about the tools that are being used in the clinic to further understand the health of humans and diseases?
Yeah, so the immune system, I have this like saying when I was like learning autoimmunity and rheumatology, that it's like studying like Star Wars or something. It's just so complex. And if it's involved in pretty much every disease process, either all infectious diseases. It is kind of this dance between the agent and the host, which is the immune system. Now we know, like, it's so relevant for cancer. It's just the system that really will shape how you will face the environment in all its ways, starting with the microbiome. That is actually such an exciting new avenue of research, but the microbiome itself is hosted, by the allowance of the immune system. So it really is, it's the immune system is pretty much involved, I think, in every areas of health and disease as well. And I think the more that we can understand it, the more that overall we will understand all diseases. And it's a very challenging system to actually profile in the clinic to really understand what's going on. I think we have a lot of parameters that we can measure to see if there's inflammation and there's systemic inflammation. We can measure antibodies and autoantibodies in serum. There's other tools that we can use in clinic, for example, just how much circulating blood cells that you have, and that can tell us a lot about different populations of immune cells that are in your bloodstream. And you can do a little bit further steps now with flow cytometry to actually detect specific cell populations that might be circulating in the blood at that point when you see a patient. But overall in the clinic, it's not that we have like a huge amount of tools to really understand at a very precise level state of immunity. We can also probe the immune system in the sense of, like, if we give you a vaccine, we can see, well, did you actually develop memory? And then there's a lot of panels that we can do to actually test whether you actually really responded to vaccines that you've gotten in the past, and then probe how that arm of your immune system is. And then there's a lot of things we can also measure in terms of, like, the immune system have all these like molecules for communication that we call cytokines and chemokines. And there's a few that we can measure. Their interpretation sometimes is really hard right now in a clinical setting. So I think we have a broad sense of, if there's a disturbance of in the immune system, we can say that there's immune activation. We can say that you have like a over-immune activation that we see in autoimmune diseases. We can say that you have immunodeficiencies if you're not fighting infections appropriately, things like that. But that's, I think, as sophisticated as we can get in the clinical setting. And then, I think research-wise, I think we have such potential other avenues to really profile the immune system. And I think the idea is to hopefully start integrating all these technologies from the bench side to the bed, as I say, like really do it translational. I think we could gain a lot of insight in the real world. But that's what I can say about how we can understand the immune system right now in the clinical setting.
Excellent, and it's good that you're able to collect samples and data from patients, because that is a need in the bench. So moving along, Phillipe. How can new technologies help us read the immune system?
Yes, I think Cristina alluded to this point, which is we have a lot of technologies that we can use. I think over the last 10 years or 15 years, we've seen the emergence of new technologies, higher throughput technologies that enable us at a fairly low cost and very high scale to look at different things, whether it's DNA or proteins, gene expression. I think Marina told us about this. So what we do specifically at Adaptive is using next-gen sequencing to do immune sequencing. So we really map the genetic diversity of our T-cell or B-cell receptors. And just to give you an idea of the scale of the information we can gather, we have about 30,000 genes, but because of the rearrangements that, for example, a T-cell receptor will have to be able to have T-cells that recognize thousands of different diseases or pathogens, we have about 100 million genes in our immune system. So that gives you an idea of the scale of the data that can be generated. And once you do this, the next challenge is to make sense of it. And I'm not even talking about a situation where you could do a multi-omics approach or merge that with clinical data. So we're really dealing with complex, large data set. And that I think machine learning and AI are the perfect tools to decipher that data. But you need to get the expertise, of course. That was the big reason why we decided to partner with Microsoft. In that area, so I think you'll see more and more convergence between people who are on the biology side, whether it's academic or industry, and people who actually know how to crunch this data, people like Marina, for example. And that enables us really to translate that data into information and insights. And this is applicable to a lot of diseases, as I said before. So I'm very hopeful that patients will benefit from that. Not only in COVID-19, but we have announced publicly, for example, that we're able to detect very early Lyme disease and better diagnose Lyme disease. We have also announced that the same approach has generated very positive and encouraging signals in Crohn's disease. So we're now talking not just about infectious diseases, but we're talking about autoimmune diseases. So we have the hope that by analyzing this vast amount of data, we're able to avoid what we call the diagnosis odyssey that a lot of patients go through, right? They go from one specialist to another, go through a lot of different tests. And it can take years or months or years to diagnose them properly, especially in autoimmune diseases that are very complex and can have many different subtypes. So that's really the hope that we have for big data. And I think we're really at this juncture when we're going from using these technologies for research to creating clinical products that will benefit both clinicians and patients.
Yeah, thank you very much for sharing that. And I liked that you also brought up the point or the need for computational scientists like Marina who could crunch all that data. So Marina, can you tell us how a scientist in your lab might analyze or crunch the data? And also, I think I heard immune repertoire. Can you explain what that means?
Yes, absolutely. So immune repertoire is, well, basically the adaptive technologies, that's what they measure. But in lay words, it's a sum of all B and T cells that are present in somebody's body. And of course, we can't measure all of them, but we can take a blood sample. And from that blood sample, we can understand what is the distribution of those B and T cells in a given person at a given time. And I will say this is one of the most complex types of data that I've worked with because there are many challenges with it. There's a lot of diversity, as Phillipe alluded to before. We can only get a small sample of the overall picture. So we try from that sample that we get from a blood sample to predict a little bit more what might be going on in the immune system as a whole. And there are many technical challenges in terms of, analyzing the actual sequencing information. But it's incredibly informative. And we've actually worked in my lab to analyze this type of data in several different contexts. One of those is in the context of autoimmunity. And Phillipe also alluded to that. And the disease that we worked on is multiple sclerosis. Multiple sclerosis is a disease where the immune system attacks specifically the myelin sheath that covers neurons. And as a result of that, the neurons lose their ability to conduct impulses, and it's a devastating condition. So patients are not able to walk very much, so incredibly impaired. So we wanted to see whether we could look at the antibodies that are present in the periphery, so in the blood of these patients with multiple sclerosis. And we wanted to see whether any of them cross the blood-brain barrier into the brain. So we sampled both peripheral blood as well as the CSF fluid, the cerebrospinal fluid, from these patients, and we wanted to track specific antibodies that cross the blood-brain barrier, with the idea that some of them might be used as a diagnostic moving forward. And this is work that was in collaboration with Christian van Boudegen at UCSF and his group, and it was actually carried out while I was working at Pfizer. We've also leveraged this technology in several other examples, one of which is organ transplant. And there we wanted to see whether we can use immune repertoire, so same technology, to measure the activation or the diversity of the immune system in patients who get kidney transplant specifically and reject the organ versus patients who get a transplant and don't reject the organ. And we were able to, again, come up with a measure after statistical analysis of this data to be able to say, oh, this patient is more likely to reject, or this patient might not be at that high of a chance of organ rejection. And that can be used to modulate immunosuppression treatment. This is before the actual procedure, before the surgery. So we could use this information to modulate the immunosuppression treatment and say maybe these patients might need a little bit more immunosuppression and these patients might be okay as they are. And actually, we've now moved forward to extending the study, some of it in collaboration with Adaptive Biotechnologies, which is super exciting. We've also used this technology to look at the interplay of the maternal and the baby immune system at birth, because again, we can use it to track these antibodies, and we can see which antibodies are passing through the placenta to be able to say, oh, maybe these moms have a higher chance of an adverse pregnancy outcome, such as preterm birth. So there's a number of different clinical scenarios where you can use this type of data because it's incredibly rich. Of course, as I mentioned, there are challenges with the analysis, but that's something that we're working on.
And Cristina, so there are more challenges that you face as a clinician, and is there anything that you could elaborate on or chime in on?
Yeah, I think as a clinician and also like a clinician researcher, I think we're all very excited about the wealth of data that's been generating and the opportunities to better understand. I think from a clinical perspective, and this is how I think most of clinicians are reading all this new data, it's still kind of like, okay, where is all this going to land? And like really what actually is going to make it through to clinical practice and clinical care? And I think there's, for me, one of the things that strikes me is that there's all these studies that are focusing on one type of data set or one type of way of profiling the immune system in one particular scenario. I think so it really is going to require a lot of orchestration and a lot of validation of all these different kind of findings that people are describing, utilizing a wealth of different omics data sets that we've been talking about. And I think at the end is how do we put all this together, how we put the numbers into of really good study designs, which are really hard to do because, at least from my point of view, lupus is not relatively rare, but it's not a very common disease too. So just to get really like large samples of patients that are studied prospectively with, good biorepositories where you can actually validate and prove and really have, the perfect study design that will answer all the questions that we have. It's really hard. So what we see is like little, kind of attempts here and there. And then for me, the challenge is how are we going to put all of this together into something that makes sense. And also, as Phillipe was alluding to really get a product that will help us in the clinical side. Really like a tool that we could use. It would be like the dream for me, especially in some of our diseases, rheumatoid arthritis, that has like now a wealth of therapies. Like we don't really know right now how, like which one to pick. And so it's like this trial and error that you have sometimes with patients where you're like in a period of 2 years going through like this Russian roulette of medications until you're like, oh, I hit the target, and you've wasted sometimes like 1.5 years of trying to get that patient on the appropriate medication, which is really challenging. I mean, you tell the patients like, okay, here we go, we'll see. And you have to give a drug the trial of like 3 to 6 months to see if it works. It's a lot of time. And then you might be 6 months with a medication that really was never going to work, and things like that. So I think, my vision for the future would be to diagnose a patient and be able to have some sort of really wonderful expression data set or immune profile kit or something that tells us, okay, anti-TNF, that's like, the medications that you should pick right away for this RA patient, and don't go through the other avenues of immune medications that you have. And I think RAs, it's something that could potentially happen relatively sooner. It's a little bit more easier to kind of not as well. It's a little bit less complex than lupus. I think lupus is a harder problem to tackle, but I think rheumatoid arthritis is a disease we would really benefit, because right now we're in this, we really don't know how to choose from all these different therapeutic agents. So that's a challenge I see with all these new technologies in terms of how we're going to really get them to where we can really use them at a day-to-day level.
Yeah, and thanks for sharing your experiences with data. And what I'm hearing is we're just not there yet, right? So there's still a lot of work to be done and a lot of collaborations to be had among different types of scientists. And Phillipe, can you tell us how do we leverage patient data for research, and what are the benefits to the patients from this type of work?
Yes, and I think Cristina is right. She's in a space that is a lot more complex than, let's say, infectious diseases like COVID. So that's why, for us, it was so encouraging to see some good results in a disease like Crohn's disease. But the way we approach it is from an industry perspective, we're very focused on developing products, right? That's what we do. Having said that, COVID-19 is a special situation, right? So I told you about the importance of studying T cells, not just antibodies, to understand the immune response to the disease. So we have a number of initiatives, both internally to either develop a better test for past infection, and we have results that will be published very soon, but that we talked about publicly, that show that our test is 97% sensitive, compared to probably 90%, around 90% for most antibody tests. So we think that we can provide people who want a better answer in terms of whether they've been exposed to the virus with a test that hopefully will be on the market before the end of the year. So really a product that will help patients and clinicians right away. We are also gathering a lot of samples from patients through our Immune Race initiative, and that data is being made publicly available. So researchers that are working on this disease can learn by looking at what T cell receptors are responding to COVID-19. And we hope that we'll have more and more samples from different regions. So we may learn very important insights in terms of how a patient in Japan or Italy versus the US respond to the disease. And we hope that it will help clinicians to continue to fine-tune the treatment protocol for these patients. We've all read about the news around the Pfizer vaccine with 90% efficacy, which is fantastic news for all of us, and we're very pleased with that. We see it as a first-generation vaccine, and we have actually begun work with several companies to use our technology to actually continue to fine-tune the development of other vaccines. So really helping them understand what they need to put in the vaccine to have not only a very strong immune response, but also a very long-lasting immune response. Because that's a key question. How long will the immunity last with the vaccine? So I think we're going to see applications of big data translate into products into the clinic that improve clinical outcome, probably first in infectious diseases and then autoimmune diseases and cancer. One project that we're very excited about that we're working with Genentech on is fully personalized cell therapies for cancer. I think we have all heard about precision medicine or companion diagnostics, where cancer patients are tested to see what kind of mutation they have so that we can know whether they will respond to a specific therapy. And really using this mutation analysis or next-gen sequencing to do therapy selection. Or ultimately, our approach with Genentech will be a fully personalized approach where we actually sequence the tumor of a cancer patient, for example, to detect what specific mutations they have, because a lot of tumors don't have the same mutations. And then based on this information, develop a TCR cell therapy personalized for this patient. So every patient will receive a slightly different treatment and with the promise of greater efficacy. So this is much longer term, but I think that's another example in another field where we hope that all this big data analysis and study of the immune system will translate into benefits, clinical benefits for both patients and clinicians.
Excellent. And I would also like to add that we are hosting a panel tomorrow focused on precision medicine in cancer, moderated by Alan Ashworth. So I strongly advise our viewers to check that panel out too, as they will further elaborate on Phillipe's point. So moving along, I'd love to hear Marina's perspective in terms of developing computational integrative methods and applying these approaches in the context of disease diagnostics and therapeutics. Marina, can you share your experience?
Yes, absolutely. So in terms of benefits to the patients, as Cristina was alluding to, we're getting there. And that's really our goal to ask questions that will impact clinical practice and what clinicians decide to do, both in terms of figuring out how to diagnose disease better, but also how to come up with these precise treatments. And actually, Cristina and I are working closely together on one of the projects relating to lupus. And I've mentioned before using public data, right? Data that's already out there that scientists might have put in some database, and we're trying to make sense of it. But the flip side of it is really looking at a well-curated patient cohort. And that's one of the things that Cristina, in collaboration with a number of other rheumatologists at UCSF, have done for lupus. There is a cohort of about 400 or 500, Cristina knows the numbers much better than me, lupus patients that they followed throughout time with incredible clinical phenotyping, which means all the clinical information from when these patients go to the doctor, the criteria, the lupus criteria that Cristina was mentioning about, but much more than that. And in addition to that, there's molecular data that's tied to it. So I talked about gene expression. We don't have immune repertoire, but hopefully, maybe that'll come soon. But we have many other types of molecular measurements to characterize the genetics, the epigenetics, which is the genetic modifications, characterizing the expression, characterizing environmental exposure. So all of these layers and layers of information, in addition to incredible clinical data. And that's where we can actually start studying disease heterogeneity. And that's exactly what we're doing. And sort of figuring out how to integrate these molecular measurements together with clinical data, or you can think of clinical data is any data that is captured when you go see a doctor. So it all goes in the electronic medical record. And of course, the goal of these records is for clinicians to keep that information, also a little bit for billing purposes, so that your insurance companies can get billed. But at the same time, it also provides an incredible opportunity for computational scientists to do analyses on those clinical data. These are the biggest experiments And the most expensive, I wouldn't call them experiments, but the most expensive types of data that exist. And it's a shame not to use it to understand disease better, to understand different treatment options and the effects that those treatments have on patients. And taking that clinical information and putting it together with molecular data can be, or at least my hope, can get us a little bit closer to precision medicine, to figuring out which treatments might work for which subgroups of patients. And again, I think of it as guiding hypotheses, which of course then need to be tested either clinically or in the wet laboratory. So that's the type of work that my group focuses on. And I'm incredibly, incredibly lucky to be working with people like Cristina to answer some of these questions.
Yes, thank you, Marina, for I haven't actually spoken about our study, and it's a great opportunity to also just, really put into perspective how hard these things are. So I work with several outstanding clinician investigators at the Division of Rheumatology, and we are part of what we call the CLUE study, which is the California Lupus Epidemiologic Study, and it's a longitudinal cohort of patients with lupus that come where they spend hours with us, and we really do deep interviews about like their social determinants of health. We do a physical exam, we review their medical records, we get clinical data, and we also have a biorepository of serum, plasma, PBMCs, which are the immune cells that are circulating in blood, as well as DNA, and we are generating all these large data sets with gene expression, DNA methylation, as epigenetics. And I think Marina is key right now because it's really this new approach of trying to integrate all these levels of data to really try to make sense of, to quote the name of the panel, who is the patient, and really how can we better understand that person in front of us in a personalized way, and we really it's so labor intensive and so much work to do these kind of longitudinal repositories. You have no idea how hard it is, and we're thankful for our funding sources that have made this possible, because it really does require a village. So yeah.
Wow. Okay, great. So we're almost out of time, but I would like to ask. I think we might have time for one more topic. Phillipe, just considering what Cristina and Marina were sharing, how do we move new technology into the clinic? What would you suggest?
Well, I think when it comes to the use of big data, I think it's pretty similar to what we're doing in life sciences in other fields, right? It's first, I think it's strong collaboration between academia and industry. A lot of innovation comes from academia. If I look at our company or scientific founder, Arlan Robbins, worked for many years at the Fred Hutch Institute. And we have very strong collaborations with a lot of academics in different fields. So I think we need to continue to nurture that collaboration between academia and industry. And then I think industry has a big role to play in terms of when it comes to translating this scientific innovation into actual products, right? We're used to that. And it's not unlike what we're doing in therapeutics or maybe more traditional diagnostics or medical devices. But when it comes to the use of big data, machine learning, and AI, the first step is to get a signal. So if we think, for example, about reading the immune system to diagnose a disease better or earlier, we typically analyze blood samples from cohorts of healthy patients as well as patients who have the disease, and that's where access to these large cohorts, well-characterized cohorts, can take a lot of time and effort, and it doesn't come cheap. And then once we have this analysis, we're able to see whether we see a signal. Do we see a group of T-cell receptors that are clearly responding to that disease and then can be used in patients whose status is unknown to actually confirm that they have that disease? Do we have Crohn's disease? Do we have UC or other celiac disease or another inflammatory bowel disease? because ultimately that's what we want. We want to give physicians a test where from a simple blood test, they can roll in a large number, roll out a large number of diseases and roll in and confirm the diagnosis. And once we have that signal, then we need to develop what we call a clinical grade model or algorithm. So there's more validation to do. And then we enter clinical studies like we would with a traditional diagnostic product or therapeutic, and going through the regulatory approval that is relevant in each country. So this is really, I think, the bread and butter of industry, which is turning innovation into a product. I think the front end with big data and getting a signal and confirming a signal is a little bit different. But the back end is the same, and it's there to ensure that what we do is scientifically sound, and the product is efficacious and safe for patients.
Thank you. And Marina, did you want to add more to that from your point of view with bioinformatics?
Sure. So I think I completely agree with what Phillipe said. I think collaboration, but beyond academia and industry, but also with our clinical partners is incredibly, incredibly important. I think the lines between academia and industry in general, especially in the Bay Area, I would say, have blurred tremendously. I myself have a very non-traditional academic career path. I actually worked at a pharma company for a few years before joining UCSF. I was at Pfizer to better understand the process of drug development and how that happens before coming back to UCSF to lead a research group here. And I see that happening more and more. Many faculty are involved with startups, or many of us are funded by pharma companies to do our work. So I think this collaborative environment of being able to take ideas, not be afraid to work with an industry partner to move those ideas and products forward, because really that's what they're good at. And we're good at doing research and kind of trying new crazy things. And there are people in the industry who are really good at figuring out which of those things can make a good product or a good drug. So that's something that is very important to keep in mind. But at the same time, for me, as I'm not a clinician, for me to understand what the important questions are, even before starting a research project. So talking to somebody like Cristina and understanding what are the big problems in lupus, what are we facing, what do we need to solve, and then figuring out, does the data exist to solve those questions? If it does, great, let's try that using public data or something that is already there. And if it doesn't exist, let's generate those data together and try to address those questions. So I really think that it's important to be able to collaborate and talk to each other. And communication is incredibly important, because we all speak different languages, right? Whether we're in academia or whether I'm talking to a clinician or a computer scientist or a biologist or a business person in the industry, I need to figure out, how do I talk about what I want to do or what my background is or what the methods are in a way that everybody understands. And I really think that that is something that can help us move research forward, and hopefully, not only use patient data, but also help patients in terms of diagnostics and therapeutics.
Excellent. And yes, Cristina, so, to add to Marina's point, we have an industry expert, computational scientist, and you, the doctor. That almost sounds like an opening to a joke, but how do we continue this conversation? How do we keep this going on so that we know, the right questions to ask?
Yeah, no, I echo everything that Marina said. And one of the reasons I'm still a clinician, like I keep my panel, my patient panel, instead of just jumping off to research, it's because really, like, that really concrete way of just seeing the problems in the clinic and just learning from our patients, it's what really drives the research questions, and also brings the world of research that can be really super exciting, but it really lands it into like, okay, something that we really need to do at a practical level instead of just like wondering about like musings of biology and life and sciences. And I think really, I really do echo everything that has been said here. And I think also the devil's in the details too, and a good study design really needs a clinician to really kind of really put into perspective what needs to be answered, and if that is actually going to be relevant. So I really think that this is kind of like the way that I think we should be going forward. Good clinical insight, great computational tools, and someone that delivers a product, which is, I think, a great kind of summary of how we should move forward with these research studies.
Okay, everyone. Well, we are officially out of time. Before we go, I would like to thank the scientific department of the Embassy of France, Cristina, Phillipe, and Marina for joining us. And many thanks to the viewers for watching the panel. I hope we gave you a better understanding of the importance of collaborations among basic scientists, clinicians, and industry business developers across the universe. From San Francisco to Paris, I'm Gina, and I'm proud to be a contributor of the Quantitative Biosciences Institute, an organized research institute under the School of Pharmacy at UCSF. Thank you all.
Thank you.
Thanks.
Thank you.